Good afternoon, everyone. On behalf of the Center for Scientific Research and Development, People's University, Bhopal, I welcome you all to another exciting edition of the BioConnect webinar series, a platform intended for students by students to ignite curiosity, foster dialogue, and bring real-world relevance to academic insight. My name is Tarun Visvakarma, and I'm proud to tell you that I am a Biotech degree program student at CSRD. I mean, the Center for Scientific Research and Development at People's University. It is my honor to serve you as your MC for this session. Today's theme is truly exciting, transforming ideas into products, commercialization in the biotechnology era. We are deeply honored to have the several distinguished personalities joining us today. We welcome Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, our esteemed speaker from Malaysia, Dr. Harikrishna Kulaveerasingam, former Chief Research and Development Officer, SD Guthrie Berhad, Petaling, Jaya, Malaysia. Head of the Center for Scientific Research and Development for this initiative, Professor Dr. Subhash J. Bhore, my fellow student and chairperson of the organizing committee, Ms. Muskan Chandral, and most importantly, participants from India and beyond. Thank you all for your presence. It truly enriches this session. To formally begin today's webinar, I invite the Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, to deliver opening remarks. Sir, over to you.
Thank you, Tarun. Good afternoon, everyone. It gives me great pleasure to warmly welcome you all to this very special session of the BioConnect webinar series, hosted by our enthusiastic and talented biotechnology students at the Center for Scientific Research and Development under People's University, Bhopal. Today's theme is transforming ideas into products, commercialization in biotechnological era. This is both timely and inspiring. In a world that is rapidly shaped by innovation, the ability to transform creative ideas into impactful solutions holds the key to progress, not just for science, but for society and the economy at large. We are deeply honored to have with us Dr. Harikrishna Kulaveerasingam, former Chief Research and Development Officer at the SD Guthrie, Berhad, Malaysia, a distinguished leader who has dedicated his career to advancing biotechnology through research, innovation, and commercialization. Sir, your presence here today is indeed a privilege for all of us, and we eagerly look forward to your insights and wisdom. What makes today's program truly special is that it is student-led. A dedicated team of 6 biotechnology students from the Center of Scientific Research and Development have conceptualized, designed, and organized this webinar under the guidance of their mentor, Professor Dr. Subhash J. Bhore, head of the Center for Scientific Research and Development at People's University. This reflects the spirit of for the students, by the students, an approach that not only builds confidence and leadership amongst our young scholars, but also creates a culture of innovation and ownership. Dear students, I want to take a moment to congratulate you. By organizing this session, you are not only connected with the global thought leaders, but also building bridges between academic learning and real-world application. This is exactly the kind of initiative that strengthens our vision at People's University to nurture talent encourage creativity, and empower our students to become leaders of tomorrow. As we begin today's session, I encourage all participants to engage actively, ask questions, and take full advantage of this golden opportunity to learn from one of the finest minds in biotechnology. With that, I once again welcome Dr. Harikrishna, our esteemed participant and our student of NIDER. I wish their webinar great success and look forward to the inspiring discussion ahead. Now I will hand it over to our MC. Tarun, over to you. Thank you.
Thank you, sir, for your encouraging and inspiring words. Now, I invite the chairperson of the organizing committee, Ms. Muskan Chandrol, to say a few words. Over to you, Ms. Muskan.
Thank you, Tarun. Hello, everyone. Our esteemed guest speaker, Dr. Harikrishna Kulaveerasingam, our Honorable Vice Chancellor of People's University, Professor Dr. Harikrishna, our mentor, Professor Dr. Subhash Bore, respected dignitaries, respected faculty members, and dear fellow students. I welcome you all to this session of the webinar. My name is Muskan Chandrol, and I am proud to tell you that I am a BSc Biotechnology student at the Center for Scientific Research and Development, People's University, Bhopal, India. It is a true honor to serve as the chairperson of the organizing committee of this program. On behalf of the organizing committee, I warmly welcome all of you to this thought-provoking webinar titled Transforming Ideas into Products, Commercialization in the Biotechnology Era. The biotechnology industry is witnessing a golden era. The groundbreaking ideas are rapidly evolving into market-ready products. Today's webinar focusing on transforming ideas into products, commercialization in the biotechnology era. We'll explore the journey from innovating research to successful commercial ventures, featuring insight on strategies for navigating intellectual property, securing funding, meeting regulatory requirements, and scaling production. I'm sure that at the end of this session, you will have an understanding of how visionary concept in biotechnology can be transformed into products or services that drive scientific and industrial progress. On behalf of organizing team, I am honored to welcome our esteemed guest speaker, Dr. Harikrishna Kulaveerasingam from Malaysia to our BioConnect webinar series. I'm confident that our guest speaker thoughts will help all of us to understand better what it takes to transform ideas into products and their commercialization. With these words, I thank you all once again for joining us. Let us look forward to an inspiring and insightful session ahead. Now, I hand it over to MC Mr. Tarun. Over to you. Thank you.
Thank you very much, Ms. Muskan, for your welcoming remarks. Fellow students and esteemed participants, we are now ready to begin the core session of our webinar. Now, I hand over the session to our moderator, Ms. Kirti Vaswani, to introduce the speaker and to moderate the session. Ms. Kirti, over to you.
Thank you, Tarun. Hello, everyone. A very warm welcome to all participants joining us from across India and around the globe. I am Kirti Vaswani and I am proud to tell you that I am a BSc Biotechnology student at CSRD People's University, Bhopal, India. It is my privilege to serve as your moderator for today's insightful session organized under the BioConnect webinar series, an appreciable initiative of the Center for Scientific Research and Development, People's University. Today's session focuses on a topic of a significant relevance in today's fast evolving scientific landscape: transforming ideas into products, commercialization in the biotechnology era. We are living in an age where science and innovation are not just transforming industries but shaping every fabric of our society. In biotechnology, an idea that is born in a lab has a potential to become a life-saving drug, a sustainable food source, or an eco-friendly industrial process. Today, we will explore exactly how we bridge that gap between the bench and marketplace. To lead us through this crucial discussion, we are honored to have with us an eminent scientist, Dr. Harikrishna Kulaveerasingam. Dr. Harikrishna, who served as the Chief Research and Development Officer at SG Guthrie Berhad, Malaysia. He has led the impactful research and innovation programs in biotechnology, turning scientific ideas into commercially successful solutions. He has spent decades leading the biotechnology research, from developing innovative solutions in sustainable agriculture to guiding products from lab to marketplace. Dr. Harikrishna is well known for combining expertise with a clear focus on practical applications that benefit both industry and society. His journey is an inspiring example of how ideas can be transformed into impactful, genuine world innovations. With this very brief introduction, it is my honor to invite Dr. Hari Krishna to deliver the talk on transforming ideas into products, commercialization in biotechnology era. Dr. Hari Krishna, over to you.
Thank you very much for the very kind introduction and from your Vice Chancellor, as well as your organizing committee. I'd like to thank Dr. Subhash as well for inviting me to share some of my life work with you guys. Hopefully you don't fall asleep over the next 40 minutes or 45 minutes. Okay, so let me pull up my slides. The title given to me is Transforming Ideas into Products and Commercialization in the Biotech Era. I'm going to tell you a story about how we have essentially done fundamental research and converted it into, let's say, some commercializable tools. However, it's taken a few years to do it. So, firstly, I would define what commercialization of innovation or biotechnology or R&D is. I'll give you some introduction to oil palm. Then I'll tell you a bit about our story and also about some of the people who were involved, who were instrumental in the success of this program. And then finish off with proof of the pudding, which is validation of the performance of these materials in the field, right? And then what were my key learnings from this whole program? And then we will go for question and answer. So, commercializing R&D discoveries. Really, this is translational research, right? So it involves translating scientific discoveries or innovations into marketable products or services. Now, this is really difficult to do because it has many touch points. It is multifaceted. It includes many different areas of legal, science, and so on and so forth, such as securing intellectual property rights, developing a robust business plan, securing funding, navigating regulatory pathways, and effectively managing the product life cycle. So, a bit about the oil palm industry. It's in desperate need for innovation, and over the course of the next few minutes, you will understand why. This is the oil palm here, and those are the bunches close up. So oil palm actually originated in West Africa, and it was used for various purposes over thousands of years. It essentially grows in the tropical belt of between 5 to 7 degrees north and south of the equator. So it requires quite a lot of water. It was first sort of commercialized in the modern world, let's say in the last century, by Lever brothers, who started to use palm oil as a means to make soaps as well as candles. And then given that many countries were colonized at that point in time, and that both the Netherlands and the United Kingdom essentially had many colonies, so they expanded oil palm production to their colonies, to Indonesia and Malaya at that point in time. So they could actually produce and consume at least reasonable amounts of this particular product. Now, palm grows in the tropical biome, and I have this up here because the tropics actually host more than 50% of all vertebrate species and more than 70% of all flowering plant families. It also gets a huge amount of rainfall. 40% of the global rainfall and its recycling happens in the tropics. And therefore, it is a very sensitive region with regards to maintaining that forest cover, and also with regards to the way in which the public around the world view deforestation, right? So this is one reason why palm oil or oil palm has been vilified for many years, because many people see its expansion as an expansion into these forested areas and this destruction of these rainforests. However, having said that, many companies, most companies today have an NDPE objective, which is no peat, no deforestation, and no exploitation of the indigenous people. So they have this as part of their commitment for oil palm growing. So why is oil palm important? It is important because it is the most productive oil crop in the world. If you look at this part of the graph here, this bar chart, you can see that this is average yield. So there could be much higher yields with specific companies or specific countries, but this is average global yield, and it's fourfold higher than the next highest oil yielding oil crop, which is sunflower, and 8 to 10 times higher than soybean. But it's only planted on 8.5% of the land area that has been put aside for oil crops, but produces 30%, 36% of the global oils in the marketplace. So it punches above its weight. Another reason why palm oil is important is the fact that it is produced year-round. It's not just a summer crop. And it has chemical versatility, which ends up as it being used in a wide range of different products. And the reason for this is that if you look at the fruit, cut palm fruit here, you have the mesocarp, fleshy mesocarp, and you have the kernel in the center. The kernel essentially produces kernel oil, which is very similar to coconut oil. It is more saturated and is used for industrial products such as soaps and other oleochemical products. Whereas the mesocarp is less saturated, and this is used to make vegetable oils that are used for frying. And palm oil has very high stability characteristics, which means that it is used in many industrial products. So soup noodles, noodles, biscuits, and cookies and cakes and so on and so forth, pie pastry and so on and so forth. Now, there's a huge demand for palm oil, and it is estimated that by 2050, the world needs to double its production. That means in 25 years, we need to double our production. And this needs to be done without expanding land area for cultivation, right? So the only way out is to actually innovate and come out with superior yields and production, right? Now, this demand is driven by increased demand in India as well as in Africa. Now, the story in Malaysia is slightly different. You can see this bar chart here that the yield, the oil production has sort of stagnated to a certain extent. But the yield on a per hectare basis has been declining for many years. So given that land expansion is not an option, then replanting with superior planting materials is the main option moving forward. And improvement in productivity is essential to ensure that we are able to continue to supply the market with cost-effective palm oil. Okay, this is a picture of a scientist called J. Craig Venter. Now, why have I put his picture here? It is because in the early 2000s, so late 1990s and early 2000s, there's improvement in sequencing technology as well as in a field called bioinformatics or computational biology, right? Computational systems, which allowed the sequencing, large-scale sequencing, and assembly of genomes to be achieved with at least be achieved reasonably successfully. So in 2003, Dr. Venter and Dr. Francis Collins actually completed the human genome. The rice genome was sequenced in 2004, and Craig Venter formed this company called Synthetic Genomics with the aim of ultimately producing synthetic life. So the idea is that if you can remove all non-essential functions and only put in place the functions that are required for an organism to produce a specific product, let's say a specific type of molecule, then that organism can push all its carbon towards producing that kind of molecule and thereby being extremely effective and efficient. However, this dream is still ongoing and it's not been successfully commercialized yet. But in 2007, Synthetic Genomics and a Malaysian company called Genting, through its subsidiary called Asiatic Center for Genome Technology, or ACGT, formed a collaboration to sequence the oil palm genome. And when I met with their team to discuss collaboration, the terms of collaboration were very unfavorable to, at that time, Sime Darby plantation. So when I told my management about this, they were very concerned and they decided that we needed to actually move ahead on our own to actually do the genome. So the first thing that was required was actually to secure support, which I already obtained. I got the management buy-in that we should do this project. And in doing so, I secured commitment for long-term funding, also secured commitment for expanding the research team to actually get the work done. But having said that, at that point in time, I was the only individual in the company who actually understood this technology. And therefore, I felt the need to develop a robust project governance and oversight structure. So we established an independent scientific advisory committee that was essential to our project success. Then together with them, we started to build the R&D pipeline and try to identify potentially commercially relevant products. So let me share our SAC with you, because I think this group of people were essential or very important in ensuring that the project was successful. So we had a scientific advisory committee as well as a set of consultants. The scientific advisory committee met on a 6-monthly basis, whereas the consultants actually interact with the research team on a, in some cases, daily to weekly basis, right? So what is special about the SAC is the fact that all of them have industry experience. And Prof. Sinsky, who was the chairman of the SAC, had commercialized $3 billion companies from his research program at MIT. So these were people who actually knew how to convert science into products. So I'd like to actually pay some tribute to my SEC members who have since passed on. This project has taken a long time to happen. So Prof. Sinsky basically has passed away last year. And some of these statements here are his sayings, right? Stay between the yellow lines, meaning to say don't go off the road. Time is money. The honeymoon is over, meaning to say you need to deliver value. What did you learn today? We always keep asking ourselves the question, what did we learn? Keep it moving. So don't stagnate. And the pitch formula for success, meaning to say form small teams that are dedicated and resilient and resourceful and move it forward. The other SAC members who've also passed on, Professor Mani Subramaniam, who told us don't use a laser to kill mosquitoes. And this is especially pertinent for scientists because we tend to overcomplicate issues. So in other words, try and keep things as simple as possible and develop the relevant technologies for the relevant problems. And Professor Andy Greenland, who kept telling me, this is great science, Harry, but where is the commercial product? So we are always under pressure from these gentlemen. And Dr. Herwood Cawley is the father of oil palm genetics, and he's written the book on oil palm genetics, or oil palm, called The Oil Palm. And he, in 2005, had proposed that genetic authenticity of oil palm breeding and seed production was essential to ensure that yields were maintained. But the technology didn't exist at that point in time. But over a period of time working together, we had developed that technology, and he was very happy to see it actually in action. So we got the funding. We have now a program that needs to be moved forward. We have the genome. So the question is, what is the target? So Prof. Sinsky always told me, identify class A problem, meaning to say something that is extremely difficult to do or extremely difficult to crack. But if you did crack it, it will give you the biggest value, or it will give you significant value, should I say. So we decided that oil palm yield is the key target. The reason being that it's multigenic. It is also, the outcome is also influenced by the environment, and therefore it's highly complex, and it's not easy to get a handle on it. So we, together with our SAC, we developed an R&D strategy to unlock these genetic components of yield. And then it was a bit easier to come up with a commercialization strategy. So we decided the easiest thing to do was to actually incorporate the technology into improved seeds, because that would provide a simple, scalable solution. And we had a seed sales unit that would be the route to market for these products. So we at SD Guthrie or Samdabi Plantation, we had certain advantages, one of which we had been conducting plant breeding since the 1950s on oil palm, and there was a huge amount of both planting materials as well as breeding trial data. We also had an ongoing large breeding program with advanced planting materials that showed a large distribution in yield. So given this, we decided to leverage on these materials, and we adopted two complementary approaches. One is a highly scientific integrative omics approach, and the other one was based more on population genetics. So maybe I'll describe a bit about this integrative omics approach. So in this population of palms, they were segregating, despite the fact that they are sibs, full sibs or half sibs. That means they were related, very closely related. there were individuals that were low yielding, and there were individuals that were very high yielding. And there was a significant difference between the two in terms of the yield profile. So the lower yielding palms were yielding something like 7 tons of oil, whereas the high yielding palms were delivering in excess of 11 tons of oil. So we identified 21 individuals that could be categorized into this low yield or high yield. And then we collected fruit bunches at different stages of development and subjected it to the various omics technologies. That means transcriptomics, proteomics, as well as metabolomics. And then integrated the data onto the KEGG pathway to actually identify some of the key rate limiting steps in the production of oil. And in doing so, we have identified in that population some of the rate limiting steps and the reasons why the flux, the carbon flux was not going into oil, but being actually consumed by other pathways. Then the population genetics approach essentially uses genetic variations by our technology to be identified and then linked with the traits and how those traits segregate in various populations and crosses. So as we started doing this work, we then realized we needed to protect our knowledge and our whatever innovation that we had uncovered, right, or developed. So we wanted to provide the company with freedom to operate and also erect barriers for entry for anybody else to compete with us. And then by doing so, we hope to actually provide the company a competitive advantage in that space. So we developed this IP strategy that supported that. Part of that strategy required our team to actually understand what is required for a patent and educating them on what is considered to be novel and what could be actually patented. We also establish a formal structure for managing IPs at SD Guthrie. Now, when you file for patents, they're quite expensive to maintain. So and the bill ends up being tens of thousands to the millions. So we have developed a structure whereby all of these IPs are actually reviewed every several months to see whether there's still value in it or not, and should we still continue to protect it or to actually allow it to lapse. And in doing so, we maximize value of those IPs. We also establish a formal structure within R&D for evaluating what inventions should be IP, and what kind of IP protection should be provided to those inventions or knowledge. For example, you can't patent sequence, but you can actually get a copyright for DNA sequence. So we've done all of this in line with our IP strategy, right? Then, given that we were the leading company actually doing this kind of work, there was so much work that needed to be done to really move the industry forward. So we wanted to actually share information that did not conflict with our IP strategy with the general public. And so we also then started to come out with a strategy to publish some of our findings in scientific literature so that other companies and universities and individuals could also innovate and actually help to move this field forward. So this was sort of the journey from 2009 to today. So in 2009, we had the first sequence. So as you do know, DNA sequence is just a series of letters. But at that point in time, we didn't know what it did. And then from that large commercial breeding program, we released in 2010 a new product called Calyx 600, which is the advanced planting material. And we worked on that population using our population genetics tools to actually provide an understanding of those yield components. So in 2013, the Malaysian Palm Oil Board actually published and shared their oil palm genome bill. And they also published some seminal papers in Nature on single gene traits that were important, things like shell, and then virescence for the fruit color, and karma, what they call karma, which is essentially leading to fruit abnormalities in the tissue cultured lines. So working for a commercial company, one has to deliver some commercial outcome and you have to share with your management a plan to deliver that commercial outcome. So in 7 years, we have managed to actually convert, let's say, the DNA sequence into knowledge, and then to, with that knowledge, actually develop tools that allow us to come out with a commercial product that could be planted in 2016. And I will tell you a bit more about it shortly. Then in 2019, we had the first commercial harvest from that. And we also published an improved version of the genome, which was about almost 90% improved over the first genome build by Malaysian Palm Oil Board. So this was published so that everybody can use it. And then since then, we have been planting in various locations, our so-called genome select materials. And in order to allow the company to maximize the benefit from this technology, we have shared it with our sister companies in Indonesia as well as in Papua New Guinea. So we have been replanting all our palm fields that have been felled for replanting every 25 to 30 years, 20 to 30 years, we will replant the oil palm fields because these palms get too tall to be harvested. And therefore, by 2024 or last year, all our fields were being replanted with these materials. So this was not all done by us on our own, but in collaboration with many different universities and technology companies, some of which are listed here. So as I mentioned earlier, we needed to make sense of the oil palm genome. So let me tell you in a bit more detail what actually, how we actually went about it. So we had a reference genome that was completed in May 2009. Then we sequenced over 350 palms from diverse origins and compared it with the reference genome. So in doing so, we identified more than 650,000 unique SNPs. These are single nucleotide polymorphisms that now provided us with potential tools that we could use to interrogate genomes. Then we manufactured 200,000 SNP feature DNA chip. And then this was used to study many different populations, right? So in oil palm, these materials are called BPROs, or breeding populations of restricted origins, right? So we use these DNA chips to actually study the genetic variation across these kind of populations. At that point in time, sequencing was just too expensive to be deployed at large scale, and this was considerably more cost effective. So we also use this to study our commercial population, that Calyx 600 population, and also then to develop a potential use case for commercialization. So this is what we found down here. This is we genotype over 7,000 palms of diverse origins. So the Nigerian Everos Nigerian is wild material, and the Everos is advanced material. So this is 50% wild, 50% advanced material. And you can see it's very different from all the other materials that we have. Right? It seems to be unique, a lot of different variations in it. This is the commercial material that we are planting, the Delhi Everos. And this has been maybe gone through 3 to 4 breeding cycles. Then the Delhi germplasm is different, more variation, different variation in it. And this other dumpy Everos, which is semi-advanced material, but not as advanced as the Delhi Everos, also appears to be unique in many different manners. So I go back. So we can actually separate these different populations and identify them. And I don't know whether you know about linkage disequilibrium, but in rice and millets and other crops that have gone through many breeding cycles, the linkage LD would be fairly large, right? Meaning to say, these large blocks of DNA are inherited and in that manner, right? Before you find, let's say, crossover events or variations that have not been selected. So you can see that with a lot of selection, the LD decay becomes much, much larger. So the wild materials end up actually having small fragments, which are then which then have lots of polymorphisms in it, whereas the improved materials would end up actually having much larger LD decay. But in this case, not considerably larger, because oil palm has only gone through 3 breeding cycles since its wild days. So with our 200,000 SNP chip, we found that we had 90,000 SNPs that actually provided us with data. And we had a mapping density of about 8 kb, which meant that we could actually interrogate the whole genome. Then we went about doing a genome-wide association study across this very large population of palms, and actually look at the components which are associated with yield. And you can find, as you can see here, this is the more wild material, the Nigerian Everest. So there's something here in chromosome 5, and maybe something up here and here. Whereas in the advanced materials, you can see that the selection has resulted in a lot more homogeneity in this region here on chromosome 5, meaning to say this is one of the drivers of yield, and perhaps chromosome 11. Okay, so we then use a deep learning and machine learning algorithm to actually start to do genomic selection or genomic modeling. And this was done on this advanced breeding population that had resulted in our Calyx 600 product. So we used a training set of 60% and a validation set of 40%. And this is done iteratively, right, over many times. And eventually we got to a prediction accuracy of 0.65, which I thought was excellent, because this material is growing outside over many years, right? The yield data is over 4 to 5 years. And when we simulated actually using these markers to select for, let's say, superior planting materials, we found that in the green up here, we found that if we pick for the top 30% in the population, we'd increase oil yield by over 16%. over the mean, right? And we also determined that we required less than about 50 SNPs to actually achieve this. Today, of course, we've reduced that number because the number of SNPs that you use to do this assay increases cost. So we've got it down to a very small number of SNPs to actually do this selection. So to do all of this work at such large scale required a high throughput testing lab. And this lab is ISO accredited. So we worked with a British company called LGC Genomics, and they're the largest genetic testing laboratory in Europe, in the world, I think. Anyway, in Europe, definitely. And they do all the DNA testing for the British government, for the police, and as well as for many other European countries. And they also provide other services for chemical testing. So we have a close to fully automated system of doing DNA extraction. We can do 30,000 samples a month. And then this is automated dispenser. Plates are dried in the oven. Then we can actually assay more than 5 million data points per month. Then it is based on a primer extension assay. So you have a fluorescent probe on a primer extension assay, and then this is after PCR, this is red, and then you can score, right? So you can get 5 million data points scored. So you can see the two, this is a homozygote, the other homozygote, and this is the heterozygote. So our idea was that in disaggregating population, by using the DNA profile, we could select out the top 30% to be planted, and that should provide us a yield increment of 16% based on our analysis. So we actually decided that we should do a large-scale trial of 100 hectares. So we selected at top 30%, top 50%, bottom 30%, and we planted all of these materials out in the field to validate whether these markers hold true or not, and this technology and this idea holds true or not. And after doing this, we developed the means by which we could predict which parents or which crosses produce the best progeny, and which is our Genome Select 2. And this resulted in a 95% cost reduction because obviously you don't have to genotype every single individual. Here you just genotype the parents, and then you make your crosses, and you have, with the right crosses, the right male and the right female, you get the best combination and the best outcome. So people talk about AI, and AI is a big deal today. You know, it's not alien to oil palm breeding, right? These large language models that are recently developed, biologists have been using machine learning and deep learning tools to actually interrogate genomes and other work such as that. So you can identify patterns in complex data, right? So essentially, this is how it's done. You have defining parameters, then you go through training with a massive data set to identify this guy's face, and then you figure out whether it's accurate or not, and you go through this iterative step of improving this detection technology, right? Or it means to detect. This requires a huge amount of computational power that today is available because computers are so much more powerful, and the computational power is doubling every 2 months. So when we did all this work, we also realized that we could actually come up with a tool for genetic purity, or to determine genetic purity. So we could actually ensure that all the materials that we made, all the crosses that we make, because oil palm is a hybrid, and it's a cross between a specific male palm and a specific female palm, that these were from the correct cross before they reached the customer. And this actually added value to the products that we were producing. And these are sold at a premium to customers. So this is what it looked like. So today we have more than 10,000 hectares planted in Malaysia. So in 2016, they used to go the polybags before they field planted. This is later in 2016. And then in 2018, you can see the palms are growing quite well. And in 2023, this was the same field. Palms are much larger and a huge amount of crop produced. And this is what the cut fruit looked like. So very big, thick mesocarp, thin shell, and a decent kernel, right? And this is from a different area where these materials were planted. So all looks good. Okay, proof of the pudding is in the eating. And these are the results from all those trials that we conducted. So, we predicted 16% increase in yield. So you can see that it ranges between 7 to 20%, depending on what kind of soil type and what conditions these materials have been planted in. So more or less, we validated that the technology works at scale. So we've also developed this quality testing technology. So, and we also have a ISO accredited laboratory for ensuring that all these tests are done to high standards. So the yields also are very high, as you can see, and besides oil yield, right? So actual total yield is also high. So what did I learn? What did we learn? So number one, you need to understand the business clearly and well to develop an appropriate business model, right? So in the case of what we were working on, it was a perennial crop, and that took a long time to deliver return on investment. So it was essential also to have supportive long-term funders, given that this is a long-term program. Then I think what is key to success is that you need to have independent oversight that keeps you honest and also provides comfort to the management and funders. Success is built on actually conducting high quality science. Taking fundamental research to a commercial outcome is not trivial, and therefore you can't take shortcuts. Things need to be done properly. You need to have sufficient controls, the relevant controls in your experiments, and they need to be done robustly. So it's also important to understand the basis for the biological phenomena and innovations that you have developed and are commercializing, because you really need to understand how these products do work, because at some point in time, especially when you're dealing with complex biological systems, you get unstuck. I think it's important for you to actually work from first scientific principles, rather than to be hung up with dogma. So you shouldn't be afraid to be unique. If it makes scientific sense, then you should move forward with it. Good teamwork and communication is also a key to success. This is done by a large team of people, and you need to build a winning mindset and shared values to drive success. So thank you.
Thank you so much, Dr. Harikrishna, for that insightful and inspiring talk. You have given us a deeper understanding of how biotechnology innovations can successfully make the journey from concept to commercialization. We will now proceed to the interactive Q&A session. Participants, please post your questions in the chat box or use the raise hand option if you would like to speak. While we wait for your questions, I will begin with a few to get us started. My first question is, Dr. Harikrishna, in your experience, what are the most critical steps in successfully transforming a biotechnology idea into a marketable product?
I think you need to develop uh, a superior product, right? A product that is superior to the other competing products in the marketplace, right? Both in terms of its quality, utility, and cost. So if you have all of that, then you can outcompete others, right? So you're likely to be successful. But if your product, let's say, is inferior in certain respects to other competing products, then you have an uphill battle to actually move it out into the marketplace. So unless, of course, you're developing a unique product, if you're developing a unique product, then you have different problems to deal with, because if the marketplace has not seen, let's say, for example, you have developed a television and the marketplace has never seen a television before, how do you go about selling that television to everybody when they're still using radios, right? So, then the runway to actually making it a commercial success becomes much more difficult and much longer. But if you're, let's say, coming out, let's say, with a cosmetic product, and it's a replacement for existing cosmetic product, then you need to ensure that that product actually is superior in at least some aspects, either cost competitive, better formulation, better outcome, better user feel, whatever it is, right? I hope I answered your question.
Thank you, sir. My next question is, what are the biggest challenges faced during the commercialization of biotechnology innovations?
I think the biggest issue is getting financing, right? Because quite often when you talk about biotechnology, these are all long-term programs, right? So to go from, you most probably would have developed an outcome from your research, and then it looks interesting. So you need to have that gap between, looking interesting and actually commercially deploying a product out in the marketplace. That gap needs to be funded, because if you can't find the funding, then it's not going to go anywhere, right? So that's one issue. The other issue is regulations. So especially for biotech, if you're dealing with, say, transgenic organisms, or recombinant organisms, and so on and so forth, or specific type of products, there are all kinds of regulations, depending on the country that you're operating in, or which country you're going to sell in, that would complicate life, right? So you need to understand that. Then it is also the attitude of people. Quite often, scientists don't understand business, and as Mani Subramaniam kept telling me, don't use a laser to kill a mosquito. And what it means is, scientists tend to overcomplicate life, right? They technically oriented. We love science, right? We love technology. We love all this clever stuff, right? So there's always a tendency to overcomplicate what needs to be done. And I think that is also an issue. So sometimes we need to take a step back, and look at the bigger picture, and then figure out what you really need, right? Do you really need a laser to kill a mosquito, or would a fly swat work, right? Or even the hands, right? Your kung fu, maybe your fingers, chopsticks will work. But anyway, use the appropriate technology to deal with the appropriate problem, right?
That's very insightful. Now turning to the another aspect, how can academic institutions and industries work together more effectively to accelerate the journey from lab research to product development?
I think that they have to build um, I wouldn't say infrastructure, but they have to build trust between industry and academia. And they need to create a platform where people can interact and understand each other. So if you are not speaking the same language, then it's difficult to communicate, right? And then there's misunderstandings. For example, I spent a lot of my education in the United Kingdom, and then I postdoc in the US, in California. They speak English. They don't speak the Queen's English, which I spoke, and I think in India you speak the Queen's English as well. So that they use different terms for 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 different for different things. For example, a trolley, right? A trolley for us is something that you push a car. They call it a cart, and in San Francisco, a trolley is the tram. You know what a tram is, right? It's a train that runs on the road, right? So that is a trolley in San Francisco, not something that you push along. What you push along to take your glassware from one lab to another is called a cart, you know. And then they don't have lorries; they have trucks. So there's all of these different terminology that ends up making people misunderstand. Even when you're talking the same language, you misunderstand what other people mean. So when business interacts with academia, then we have to bridge the gap between them to help them to be able to communicate better, right? And part of it is programs such as this, where you bring industry people in to give talks. Similarly, the attachments of scientists to industry. So this sort of build bridges, right, between the different sets of individuals, and really it is building an understanding of each other so that you can speak the same language. So once you start speaking the same language, then you build trust, right? You understand what each other means. Then you see that, let's say, the academics are doing really good work, rather than you thinking these guys are not doing anything worthwhile, right? Whereas we are doing difficult things in industry, trying to make money and competing against the rest of the world. So somehow or other they're like loggerheads, right? But suddenly you realize, hey, these academics are doing really good work, and there are lots of things that they're doing that potentially has commercial value. So why don't we work together to unlock that value, right? So I think that there's a journey that needs to be undertaken. And I was lucky enough to work in MIT, where industry walks in the corridors all the time, and they fund a lot of work that is done, but they do fundamental research that actually addresses a commercially relevant problem, right? So I no easy answer. Everybody is trying to work towards this, but I hope that I've answered it well enough for you.
To gain deeper insight from your perspective, how important is funding at different stages of innovation, and what strategies can young researchers adopt to attract investment?
Well, it very much depends on your product strategy, right? Okay. So, for example, at the moment, I'm an advisor to a particular startup company, and our strategy is to actually, we have a particular product that it's actually a tool, right? So we are validating that tool with potential customers, so big customers, right? So if big customers like that tool, they potentially would buy it, right? And obviously, it is not in a fully let's say, fully honed form, not fully developed and whatever else is still rough and ready, right? But we work with potential customers to see how we can improve it to fit their requirements and also understand what their requirements are, right? And in doing so, we tick some of the boxes that the venture capital guys will be looking at. Number one, is your product useful to the customer or not? Is the customer likely to buy your product? So if you're working with your customers in validating the product, they most probably would come out with some form of endorsement stating that if it does work and it does A, B, C, D, E, we are happy to buy it, right? Or whatever, right? So you go to your funders with that letter, and then your funders will be much happier, right? Rather than you scrambling around trying to find money and your product is still sitting in the lab. Right? So I think it's very much dependent on case by case basis, but that is one approach, right? That's one approach. So essentially, getting engagement with your potential customers is a good way forward. And if you are developing like a software-based system, you can actually do beta testing with potential customers outside, right? And based on the feedback, that also provides good support for that product to your potential funders, right? So your potential funders would have comfort that this actually works and people are actually interested in it. I got, let's say, thousands of inquiries of when can I get it? How much is it? This, that, the other, right? I love this tool, right? I have to have it. I hope that answers your question.
Hello, participants. Do you have any questions? If yes, please unmute yourself and ask questions.
Yeah, Dr. Hari, I have one question because one of your slide caught my attention. You said yield is declining in Malaysia. So I'm just wondering, it's because of the old plantations or the farmers are going for other crops? Is the reason why the palm oil production is going down? And another question is there any plan to bring in the genetically modified oil palm in the field? So these couple of questions I have. Yeah.
Okay. So the first question is the yield decline, and is it due to people actually planting other materials or whatever it is? Actually, it's multifaceted. Okay. So firstly, it is a reluctance to replant. Okay. Because the prices are good, so they don't want to replant. Okay. The other reason is that Malaysia is dependent on foreign labor. And after COVID, when the COVID crisis happened, all the borders were closed and we could not get foreign labor. And so this is also a reason for the declining yields, right? So those palms are getting older, plus they're not looked after properly. And then there's that yield on the palm, but they cannot harvest it because no harvesters. So it's twofold, right? It's because of the aging material as well as the fact that it's so labor intensive and they don't have enough labor to deal with it. Yeah, which is why actually having higher yielding materials and putting aside, let's say, less productive land for other use may be a better way forward, because you maximize the labor productivity and you still can produce the amount of oil that the market requires, right? So that's the first question. The second question is about so-called GM technologies or whatever it is. So in Malaysia, we have very restrictive policies to GM, but recently the government has what do you call that, re-looked at some of the policies, and they that there is now an acceptance that gene editing is an acceptable technology, right? So definitely you will see an edited palm in the ground over the next 20 years, right? Because it takes a long time to, number one, produce it, and then it takes a long time to grow, and then produce yield, and then to validate the yield or whatever traits that you put in there, and then after that to go and commercialize it, right? So with a perennial crop, this is the problem, right? With the annual crop, the timelines are much shorter. So definitely gene editing is the way to go, because it is potentially faster than going the conventional route through breeding, right? And now with climate change, potentially you may not have time to go through the breeding route and then do many back crosses and many selections, right?
Yeah.
Yeah.
Yeah. Thank you.
All right. I have few more questions. What advice would you give to biotechnology students who aspire to turn their research ideas into impactful products?
So, dear students, I would say that you go and look at the low-hanging fruit. Don't go do what I did. It takes years and lots of money. Go and look at what can go out into the marketplace within 3 to 5 years. You don't want to spend, like me, 7 years doing all this stuff and then to validate it another 5 years, right? So it's a long time. I've lost my hair waiting for all of that to happen. But I mean, I'm paid for, my salary is paid by the company. But if you have a startup, you have to get somebody to pay you, right, to do all of this. So it is not an enviable task if you take a long-term program, right? So if you're a student, I would suggest that the easiest way to move forward in this area is to go for those products that have immediate relevance in the marketplace, that don't require a huge amount of effort to produce, right? Pigments and those kind of products are easy to extract and produce, but on the other hand, the value is of course much less. Developing software, predictive software and others, those can roll out quite fast and be what do you call that, accepted by the marketplace also very quickly, right? So those may be a better way to go with regards to finding things to commercialize. Microbes as well, because they grow very fast.
All right, students and esteemed participants. That concludes with a truly enriching discussion. On behalf of our team, I would like to extend our deepest gratitude to Dr. Harikrishna Kulaveerasingam for sharing his profound insights and expertise with us today. To all our participants, thank you for your active engagement and interest. I hope today's session inspired each of us to appreciate the power of communication in shaping the future of science and society. With that, I now hand the session back to our MC. Thank you. Tarun, over to you.
Thanks a lot.
Thank you, Ms. Kirti, for moderating the session. To move forward, I now invite Ms. Risita Maurya to deliver the vote of thanks on behalf of the entire organizing committee. Risita, over to you.
Thank you, Tarun. Good evening, everyone. It is indeed an honor and a privilege to deliver the vote of thanks on behalf of the organizing team and the Center for Scientific Research and Development, People's University. As we draw the curtain on today's enriching session of the BioConnect webinar series, first and foremost, we extend our heartfelt gratitude to our distinguished guest speaker Dr. Harikrishna Kulaveerasingam for graciously taking time out of his busy schedule to be with us today and for delivering a thought-provoking presentation on transforming ideas into products, commercialization in the biotechnology era. Dr. Harikrishna, your insightful and impactful talk has not only broadened our perspectives, but also inspired us to think deeply about the opportunities in transforming our ideas into biotechnology products or services. We are truly grateful for the knowledge and the wisdom you have shared with us. We also extend our sincere thanks to the Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, for his presence and encouraging opening remarks. Sir, your continued support is highly appreciated. Our heartfelt appreciation goes out to all the respected dignitaries who joined us today. Your presence makes our events not only successful but also impactful. A special word of gratitude is reserved for our mentor and guide, Professor Dr. Subhash Bore. Sir, your vision, guidance, and unwavering support has instrumental in shaping this webinar series. Your mentorship continues to inspire and empower us. We also thank our moderator, Ms. Kirti Vaswani, for skillfully navigating the session. Our sincere thanks to the chairperson of the organizing committee, Ms. Muskan Chandrol, for her support and behind the scenes coordination. Allow me to take a moment to acknowledge the spirit behind BioConnect for the students by the Students Initiative. This initiative stands as a testament to what passionate learners can achieve when provided with a platform to explore, question, and grow. Through this series, we not only learn from global experts, but also build a vibrant academic community that thrives on collaboration and curiosity. To our ever enthusiastic audience, thank you. Your presentation, engagement, and thoughtful questions brought energy and substance to today's session. Your presence truly makes all the differences. And finally, we express our utmost sincere appreciation to the management of People's University for their unwavering support and encouragement. Your belief in student-led activities such as BioConnect webinar series enables us to dream big and achieve more. With that, I now hand the program over to our MC. Mr. Tarun, over to you.
Thank you, Ms. Rishita, for expressing our sincere gratitude to all respective guests, our expert speaker, and every participant. Fellow students and esteemed participants, before we conclude, we request that all of you please take a moment to fill out the feedback form. The link is posted in the chat box. Your feedback is important to us to enhance the quality and relevance of our future webinars. Please note that the e-certificate will be sent to those who provide feedback. The feedback link is posted in the chat box. We will wait for 2 to 3 minutes to let you complete the feedback form. We trust you have taken a moment to share your feedback form. To share your feedback, please note that your e-certificate will be sent to you at the email address you provided in the feedback form. And with that, we come to the end of another insightful session in the BioConnect webinar series. Once again, thank you. We look forward to hosting you for the next episode of the BioConnect webinar series. I am Tarun Viswakarma signing off on behalf of the CSRD student team at People's University, Bhopal. Wishing you all a pleasant and inspiring evening. Thank you.